In this video we will discuss about this GlycoRNA characterization. Till previous videos we have made a point that this GlycoRNA could be rich in salicylic acid. However, we will see here with the help of mass spec in this figure that this GlycoRNA will not only related to not only rich in salicylic acid, but its composition also include the fucosylated glycans. So how they begins their analysis is that first they used a cell lines, that is IDIDCHO. So the specialty of this cell lines is that it will inhibit the interconversion of UDP galactose, UDP glycnac to UDP galnac, and UDP glyc to UDP galactose. So first and what is the importance of this conversion? The importance of this conversion is that if there is less amount of galactose, then it will inhibit the N-glycosylation formation, and if there is less amount of this galnac, then it will inhibit O-glycosylation. So, and both of these things are limiting in this IDIDCHO cell lines. So they will first check whether there is a decrease in the GlycoRNA in this kind of mutant cell lines or not. If there is a decrease in the mutant cell lines, then it will show that this the GlycoRNA will process similar way this NNO glycosylation process. So what they find out is that in the absence of both of this galactose and galnac, there is almost nil formation of this GlycoRNA. When there is this galactose is present, then there is a formation of slight amount of this GlycoRNA. So this shows that N-glycosylation plays a role in this GlycoRNA formation, or GlycoRNA formed in similar way the N-glycosylation occurs. And when they add this galnac only, then there is not much significant amount of increase. However, when both of them are added, then there is upregulation of the signals. So this shows that N-glycosylation plays a major role compared to the O-glycosylation. So next what they did is they need to check which part of the N-glycosylation plays a role. So they used they checked the dose dependent function of this NGI. The NGI is the inhibitor that inhibits the OST function. That is the first step for N-glycosylation. So we can see that when there is no amount of this NGI, there is a high amount of GlycoRNA formation. However, with increase in the amount of NGI that inhibits the OST, there is a decrease in the amount of GlycoRNA formation. This shows that OST might play an important role for this GlycoRNA formation. So next what they did is they check with the other part of the N-glycosylation, such as this K. They use this KIF small molecules that inhibit the other part of glycosylation, and from this they can see that there is not only decrease in the amount of signal, however there is a shift of signals in the towards high molecular weight size. So this is similar to the condition in which this is similar to our previous results, in which we made a point that this has happened because of the decrease in the amount of negative charges. So in this case, what might have happened is that this inhibition, this presence of this enzyme will leads to hyposylated glycan formation that has less negative charge, and because of this they are not able to move downwards or face more restrictions because of the decrease in the potential. And so So far they have made a point that N-glycosylation plays a role in the glycoRNA formation, and they use the different enzymes and then treat the cells with biotinylated and then biotinylated the samples to check which enzyme affects the signals significantly. And when they did it with silidase, it reduced the signals completely, and when they did it with PNJSF, it reduces the signals to a great extent but not completely. However, when they use the O enzyme that affects the O glycosylation, there is almost no reduction in the signals that has been observed. So this made the points that O glycosylation plays almost no role in the glycoRNA formation. It's the N glycosylation that plays a major role. Next, they did the mass spec. Next analysis they do is the mass spec analysis. So what they did is that they take a small RNA and then treat it with PNJSF and then separate them using this PGCSP column, and then use them in the PGC, LC, MSMS column and annotate them via glyconote. And they did the same experiment with the peptide with the same cell lines. And what are the observations that they got is that first observation is that there is less variability in the glycans that they get from the small RNA compared to the glycans that they get from the peptides. And same thing has been substantiated by this PCA plot. There we can see that glycans from glycans from from the RNA are close to each other, and there is less variability, whereas glycans from the peptides are have large variability. And next they characterize the glycans using the composition, and they found out that in 293 and HeLa cells there is more fucosylated glycan in the RNA compared to the peptide, and whereas for this HeLa cells there is more sialic acid glycans on the RNA compared to the peptide. So this shows that this whole figure made a point that GlycoRNA are rich not only in sialic acid but also in fucose, and they are mainly processed via N-linked glycosylation pathway. So, and if you need to go in detail, or if you need to go in detail about the whole story of this GlycoRNA, you can either refer our previous videos or go through the reference that has been provided in the description. We will continue this story in our next videos also to further characterize this GlycoRNA. That's all for this video. Thank you for your time.